Hi, I'm Janice Geary, and I want to tell you about my PhD thesis called How the DNA Barcoding Commons Are Governed, Understanding How a Heterogeneous Global Community Shares Genetic Resources for Non-Commercial Use. I was hoping to present the results for my thesis at the 7th International Barcode of Life Conference held in Kruger National Park in South Africa. Instead, I'm bringing you this YouTube video that I filmed at my home in Edmonton, where the weather is different from South Africa. The main problem my research addresses is that while we know that sharing is good for science, it's hard for scientists. We want scientists to share their data and research outputs, but we know that there are non-technical and social challenges to actually making this happen. We know that it's difficult and it takes time out of research activities. We know that when researchers share their data, they run the risk of someone scooping them or publishing their research before they even have a chance to. We also know that by making resources freely available to anyone, you run the risk of some people using the resources without ever contributing anything themselves or free riding off of the collective efforts of others. We know this because these resources for science are a type of commons. Commons are shared resources that are managed and used by a group of people and are vulnerable to social dilemmas that impact collective action. My work on Research Commons was informed by the work of Elinor Ostrom and many others who have spent decades trying to understand how people manage and use shared resources. Early work in this field focused on natural resource commons like forests and fisheries. Research commons are non-rivalrous and therefore are vulnerable to different types of social dilemmas. Until now, most empirical efforts have focused on homogeneous research commons. That is to say, the group of people that are managing the resources are very similar to each other. But a lot of research is actually global, and global research commons include a very heterogeneous group of people. So the overall aim of my research was to inform best practices for the governance of a global heterogeneous research commons. Enter my case study, the DNA Barcode Commons. DNA barcodes are sequences of short, standardized gene regions, and they enable inexpensive identification of unknown specimens. The DNA Barcode Commons is an international effort to build a reference set of linked data, metadata, and specimens to enable a wide range of applications. While DNA barcoding is promoted as a tool to identify life on Earth, the process of creating a barcode record actually starts with a taxonomically identified specimen. While many of the technical challenges to building the DNA Barcode Commons have been worked out, the goals of the international community require sharing genetic resources across national borders. Genetic resources are any material of plant, animal, microbial, or other origins with functional units of heredity and may have actual or just potential value. There are international and national laws that determine how researchers and others can access and use genetic resources. Although this legal framework was created for a good purpose, to protect genetic resources and their associated traditional knowledge from misappropriation. Many countries have laws that set out a legal requirement to share the benefits from commercialization or use of genetic resources. As lower-income countries in the global south have been historically exploited for their genetic resources, these legal frameworks have been widely implemented in these countries. Researchers did successfully advocate for simplified measures for accessing genetic resources for non-commercial work, using the example of DNA barcoding or other bioconservation projects. So using the DNA Barcode Commons as a case study, my research sought to understand more about how heterogeneity impacts collective action and governance of a global genetic resource commons. I have 4 main research aims I'm going to describe in this video, and you can use these links to jump ahead to those different sections. Starting with Aim 1, analyze the governance of the heterogeneous DNA Barcode Commons using Ostrom's framework. I interviewed 50 key informants from 14 countries, and I reviewed organizational documents from 2 main DNA barcode organizations. I analyzed the transcripts and documents using a modified version of Ostrom's framework. I also conducted a bibliometric analysis of over 3,500 DNA barcoding publications and examined the patterns of DNA barcode record submissions for 2 examples. I'm going to highlight a few of the results, but you can download more details here. One key finding was that researchers from high and lower income countries had a very different understanding of the role of the international legal framework. As examples, a researcher from Canada said, "But I do have some serious concerns about the way that biological resources are being treated, because I have no commercial interest in biodiversity." Whereas a researcher from India said, "The CBD clearly says that you have, as a nation, a right to its unique biological resources, that these resources cannot be exploited by anybody else without proper agreement and conversation." I also found heterogeneity in the contributions to the DNA Barcode Common. For example, some people contributed biological materials, whereas others only contributed data. There are also very different levels of research infrastructure available to participants and levels of capacity to contribute to the Barcode Commons. Some participants are able to perform sequencing within their own countries or institutions, and some needed to ship specimens to foreign countries to participate. One thing that most interviewees agreed on was the value of building the DNA Barcode Commons. But there was a lack of shared values in how to achieve this common goal, and little participation by lower income country researchers and stakeholders in crafting the governance structure. Many high income country interviewees were certain that open sharing was the best way to move forward to build a Barcode Commons. They often had a belief that the natural world should be freely accessible for scientific pursuits. This group of interviewees believed strongly that open sharing actually had the greatest benefit for all participants. The negative history of resource misappropriation was often ignored or explicitly brushed off. In contrast, lower income country interviewees were often concerned about not receiving benefits from sharing their genetic resources. The benefits they talked about were scientific benefits like research infrastructure, control of domestic specimens, and publications. But JANIS, an open access database, will benefit everyone. Lower income countries can also use the data. Yes, they can use the data. Despite the fact that a large proportion of the world's biodiversity is in low income countries, only 1% of the authors in my barcoding publication database were from low income countries. In fact, over half of the publications had authors from only Western countries. If lower income country researchers aren't publishing as many barcoding papers, are they still using the Barcode Commons? To understand more about that, I downloaded barcode records for 3 mosquito genera. These genera are found globally, but are especially important in low-income countries where they spread deadly diseases like malaria and yellow fever. Of the over 12,000 mosquito barcode records that I downloaded that were also linked to a voucher specimen, only one single specimen was collected in a low-income country. Overall, my analysis of the governance structure concluded that the governance of the DNA Barcode Commons is not yet globally focused. Successful governance must account for community heterogeneity and the international legal frameworks, and will help overcome the social dilemmas and increase the use and recontribution of value-added data to the DNA Barcode Commons. Aim 2, analyze media representations of commons heterogeneity. One of the core objectives of the Eyeball Project was to examine media representations to prevent an undernourished environment for wider public discussion of the project's results. Myself and a research assistant analyzed 900 English language print media articles mentioning DNA barcoding from participating countries. Analysis demonstrated that print media coverage focused on the novelty and applications of barcoding, but failed to represent the scientific complexity, the social and political challenges, and the necessity of global participation. The lack of media coverage on the social and political implications of barcoding is one indication that these discussions are not prominent within the DNA barcoding community or its outreach efforts. Aim 3, analyze whether the rules used to govern the DNA Barcode Commons mitigate heterogeneity. Rules is a colloquial term. What I'm really looking at are institutional statements. These are constraints or opportunities to prescribe, permit, or advise actions or outcomes for participants in an action situation. These statements can be simplistic strategies, conditional norms, or rules with consequences. I use 2 theoretical frameworks to understand rules and heterogeneity. Ostrom and Crawford set out a syntax to guide empirical analysis of institutional statements called the grammar of institutions. Statements can be formal and documented, or informal, unwritten standards and expectations. Once I identified institutional statements, I used institutional logics to classify and compare them. The concept of institutional logics helps explain the influence of society on organizational behavior. Scholars can use ideal types as an empirical tool. This helps systematically categorize behavior to facilitate comparisons. I know this is a bit much for a YouTube video, but this is one of the major theoretical contributions of my thesis, and I'm forcing you to learn about it. Let me explain exactly what I did. I interviewed 35 DNA barcoders, administrators, and proponents, mostly at 2 different DNA Barcode conferences in 2011 and 2013. And using the grammar of institutions, I identified institutional statements in the transcripts of these interviews. I also identified institutional statements from organizational documents and policies. Here's what the analytical process actually looked like. I identified each instance of a barcoding behavior from the text and identified the components of the syntax. Then I compared each statement to a list of ideal types of logics I created. In this example, because the interviewee stated their behavior was driven by the expectations of the International Barcode of Life project, I categorized it as corporation. However, if the person had said, "I must do this because I'm a scientist and this is how I maintain my professional reputation," I would have categorized it as profession. I compared the logics across different source types, whether it was from a high or lower income country interviewee or from an organizational document, and across different behaviors, like sharing specimens versus accessing data. For example, high-income country interviewees had 3.1 times the odds of being categorized as corporation when they were describing their behaviors for sharing specimens. Overall, what I found was that there is heterogeneity in the logics that likely inform expectations about how others should behave in the project. But while these statements reflected the heterogeneity and indicated whether there were differences in expectations, there were no institutional statements to mitigate the effects of this lack of shared expectation. We know from previous research that lack of shared expectations can contribute to difficulties in forming the collective action necessary for building a commons. I made a list of 11 recommendations for new institutional statements. These are based on where my analysis identified gaps between the expectations of the heterogeneous community. Aim 4, make recommendations for governance of the global DNA Barcode Commons. Commons scholarship has increasingly accounted for the context of international research. My research adds a perspective on how such commons function in the context of a global heterogeneous research environment. For the DNA barcoding community, governance needs to be representative of the global community, and it needs to be specific about what behaviors are acceptable within the commons. Previously, participation in governance focused heavily on providing funding for barcoding projects. But the DNA Barcode Commons isn't made of money, it's made of genetic resources. Therefore, governance decisions can't be made only by those who are bringing funding. Governance decisions need to include those who can contribute genetic resources. Governance can't assume that all community members are going to behave in the same way, because there's evidence that they already aren't. It should account for the sociocultural context of global genetic resources and the international legal frameworks for access and benefit sharing. And governance should establish mechanisms to monitor the distribution of both benefits and risks. Thanks for watching this far. If you'd like to learn more, please check out the links in the description below for more information. And my sincere thanks to the individuals and organizations who supported both my research and me while I completed it.